What we're going to speak about now is really that movement that I began talking about earlier across the capillary wall. So we call these Starling's forces. These are forces that are acting on the capillary walls. And these are the forces that facilitate fluid movement. So fluid, along with other solids that I'll describe here in a little bit, can actually move across the capillary wall. And they're going to be moving across our two extracellular fluid compartments. So thinking all the way back to Unit 1, all the way back to AMP1, that very first week, probably the very first topic we discussed were the fluid compartments within the body. We had the intracellular fluid compartments, which are fluid, which is fluid inside the cells. And then we had that larger extracellular fluid compartment, which had two subdivisions. It had the interstitial fluid, in other words, fluid around the tissues, and then it had plasma, in other words, fluid inside our capillaries or vessels. And so when we speak about the exchange of this fluid happening across the capillary walls, it's really the movement of fluid in those two extracellular fluid compartments into the tissues or back into the plasma, into the vessels themselves. And so some of the things that can go through this membrane, I think someone brought this up earlier, so we can kind of clarify here. We've got lipophilic substances, right? We know that these are fat-soluble substances or hydrophobic substances, another way to describe that can simply diffuse, so things like oxygen, fatty steroids, and so on, carbon dioxide. And then we've got lipophobic substances, things like proteins or water-soluble substances, ions, or glucose that need some type of channel or transport in order to be transported across these membranes. We've also got exchangeable proteins, and these can move across the endothelial membrane of the capillary wall via transcytosis. And if you remember back to AMP1 here again, transcytosis was that process of endocytosis on one end, followed by exocytosis on the other end. And so we can move things in and out of the cell via these vesicles that engulf some of these substances. We want to point out here, however, that there's a really important exemption of the brain, where the brain is exempt from this rule of transporting freely across the membrane. So we talked about the blood-brain barrier, and so movement into the capillaries or into the brain tissue from the capillary beds is not as free-flowing as it is in other parts of our vasculature because of the structures that form the blood-brain barrier. Does anyone remember what those structures are? Just a quick review here. Or really just making a connection here. Anyone remember the cells that help to form that blood-brain barrier?
Astrocytes.
Okay, so the cells we talked about, oh, let's see. Excellent, perfect, thank you. I'm seeing lots of responses in the chat. So astrocytes is correct. And I'm also seeing tight junctions, which is also correct. So along with the foot processes of those astrocytes, we also have the tight junctions in between cells. And so that is what helped to create this really heavily regulated, heavily controlled environment, which is in our brain tissue, and keep that separate or protected from what's happening in the vessels in our vasculature, which, as you would imagine, kind of protects the brain from harmful substances that could be in our blood. Okay, so really important to point out that this is really another exemption of the brain from the rule that happens everywhere else. Let's talk about what happens in terms of that movement across the capillary wall. So we've been speaking about bulk flow so far in the course in terms of bulk flow throughout our entire circulatory system, throughout our entire vasculature, but we can also look at bulk flow as it applies to the movement of fluid across the capillary wall. And just like it did in our entire vasculature, here at the capillary wall, the bulk flow is also going to be dependent upon the pressure gradient, okay? So high pressure to low pressure, and that natural gradient is what drives the movement of fluid across our capillary wall. And so we want to remind ourselves here, this is protein-free plasma, right, for the most part. Proteins are too large to escape through those tiny pores. Remember, we're talking about the continuous capillaries here more predominantly. And so because protein cannot move through those capillaries, what we have is simply that plasma moving, and then the proteins are left behind. And so this gives us two general net directions of movement. The first net direction of movement is called filtration. And this is a movement that is happening out of the capillary and into the interstitial space, or the interstitial fluid space, we can call it. On the other end, there's the net movement of absorption. And absorption is when we have that fluid moving in the opposite direction. It's going now from the interstitial space back into the capillary or into the vessel, okay? So the movement that favors fluid going from the capillary out into the tissue fluid or the interstitial space is called filtration. And the net movement that favors fluid going from the interstitial space back into the capillary is what we call absorption, just to make that clear. And so the purpose of these movements of fluid, in one direction and the other, is really to help balance out our extracellular fluid compartments, or equally distribute our extracellular fluid compartments, okay? Again, that interstitial fluid and the plasma, which are our two subdivisions of the ECF. And so if you think about fluid happening, if fluid were to only move from capillary out into the interstitial fluid, which is tissue fluid, we would be perpetually swollen. We would always have this edema, which is fluid gathering in the tissues. And if fluid were moving in the opposite direction only, we would have other problems as well. So excess pressures in the capillaries and so on. And so in order to prevent that, we have movement happening to different proportions, like different amounts of pressure, but we do need to have fluid moving into and out of both of those compartments in order to distribute that extracellular fluid appropriately. Okay, so here's an illustration of some of those things we mentioned that can go through the endothelial membrane here. Oxygen, CO2, fatty acids, other fat-soluble or lipophilic substances. In terms of our charged molecules like sodium, potassium, they're going to require ion channels. In terms of things like glucose and other molecules, they may require transporters, right? So they may require some type of facilitation to get through that membrane. And then we've also got some proteins, which are exchangeable proteins that can freely move in and out of the cell via transcytosis. So in being engulfed into a vesicle, being endocytosed or exocytosed to move in or out of the cell respectively. And some examples of exchangeable proteins, just to put some context here, if you think about going to the doctor and he usually measures your cholesterol, and it's usually broken down into HDL and LDL, and those are some examples of those exchangeable proteins. So high density lipoproteins or low density lipoproteins, good cholesterol and bad cholesterol, are the proteins that help to move some of those fats around our body. And so they can move into and out of our cells freely via being engulfed within these vesicles, just as an example here. Okay. All right. I'm going to pause again before we get into the capillary forces here and see if you have any questions, and then we'll keep going. Okay. All right. So what we're going to speak about now are the forces that govern movement of fluid across the capillary wall. These are called Starling's forces. And just to give a disclaimer here, these are not in any way related to the Starling's law of the heart. They just happen to have been discovered by the same physiologist. But the Starling's forces here are really these push and pull forces that facilitate bulk flow across the capillary wall. We've got two divisions of the Starling forces. We've got the hydrostatic pressure, which is a push force, and then we've got the osmotic pressure, also called the oncotic pressure, or sometimes the colloid pressure. Those are all synonymous terms, and that's going to be a pull force that is moving across the capillary wall, okay? And I just really want to make this clear. We have these forces acting on either side. So we have a capillary hydrostatic pressure, we have an interstitial fluid hydrostatic pressure. We have a capillary osmotic pressure, we have an interstitial fluid osmotic pressure as well. So let's further describe these pressures here a little bit. The hydrostatic pressure or hydrostatic pressure gradient is due to the force exerted by the presence of liquid in that specific area. And so this is going to be a push force or a thrusting force that wants to gust or move fluid or push fluid away from where it is located, okay? On the other end, the osmotic pressure or the oncotic pressure is a force which is a pull force that is exerted by the non-permeating solutes that are present in that respective compartment. And so it's only going to be exerted because of the presence of these non-permeating solutes. The big example here are proteins. And I pointed out earlier that proteins cannot cross the capillary wall. They stay behind in the plasma. And so because they stay behind in the plasma, they exert that pull force. These are called non-permeating solutes. And so they exert that pull force on the capillary, trying to pull fluid into the space that it's coming from. Okay, we also want to think about this as the oncotic pressure or the oncotic force that is being exerted by the protein that cannot otherwise move across the capillary wall. And I'm going to kind of illustrate this here just a little bit at the bottom of this slide. Whoops. Okay. And this is going back to an example of something we talked about last semester. So if I have a container here, just to really explain this non-permeating solutes bit and the oncotic pressure. If I've got a container and it's got a semi-permeable membrane in between, in other words, a membrane that allows the movement of water, but not the movement of the solutes that are in either of these solutions. Let's say I have more of a solute dissolved on the left and less of a solute dissolved on the right? So I've got high solute concentration here, low water concentration here. On the other end, I have low solute concentration and high water concentration there, okay? In order for these two solutions to get to what we call diffusional equilibrium, what has to happen here? Bearing in mind that the solutes cannot move, what has to happen to equalize these two solutions? Anyone remember? Excellent, perfect. So I'm seeing in the chat here, water has to move, and someone is saying more specifically, water has to move from the direction where there's low solute or high water concentration to the direction where there is high solute or low water concentration. And that's perfectly correct, okay? So we have to see water move from here, so in this direction. And so we now end up with less water on this side, more water on this side, but now the solutions are at diffusional equilibrium because we have that water movement, okay? So we can say that this is now at diffusional equilibrium. And so this is the premise that we mean when we talk about these non-permeating solutes exhibiting this osmotic pull. So if we have a solute dissolved in a solution and it cannot move, so think about here, your proteins are dissolved in the blood and they cannot move across your capillary, they are going to exert that pull force that now pulls fluid from the side where there's less of the solutes into the side where those solutes are located in order to balance out the fluid compartments in either side, okay? And so as we speak about oncotic pressure or osmotic pressure or colloid pressure or that pull force, I want you to be thinking about this relationship in mind. All right.